Glands are specialized group of cells modified to perform the function of secretion. If we see the development of exocrinal glands, the cells from the surface epithelium will start proliferating. They'll proliferate deep into the mesoderm and they form cords. Now, the cells within the cord undergo apoptosis and the cord is now canalized to form ducts, and the mouth of the duct opens in surface epithelium and the other end of the duct opens in the secretory unit of the gland. So in case of exocrinal gland, there is a duct system and the gland pours its secretion into the duct. Case of endocrinal gland, the cells of the surface epithelium will proliferate and the cells will enter deep into the mesoderm. But the cellular cord which is connecting the surface epithelium with the cell mass present in the mesoderm will not canalize, and only a cluster or clump of cells is seen in the mesoderm. The base of such gland forms cellular cluster, whereas the duct is not formed, and the blood capillaries and sinusoids are abundantly surrounding such cell mass, because in case of endocrinal gland, the secretory unit will pour its secretion directly into the blood, and rest of the part, which in case of exocrinal part a gland was forming the duct, in case of endocrinal gland that part will get degenerated. So in endocrinal gland we do not have ducts, and the secretion from such glands is poured directly from the cell mass into the blood capillaries which are surrounding the cells. Important differences between exocrinal and endocrinal glands is that exocrinal glands have duct in them, whereas endocrinal glands are ductless. The exocrinal glands are large in size. Their secretory units are called acini or alveoli, and mostly the exocrinal glands are separated into lobes and lobules and they have poor blood supply, whereas endocrinal glands are mostly small in size. They form cords or clumps or clusters of cell mass. They are having rich blood supply. As they are ductless, the secretion is directly poured into the blood, and they are devoid, mostly they are devoid of lobes and lobules except thyroid gland. Then in case of exocrinal gland, the secretions are called as enzymes and they act locally, and they are regulated by autonomic nervous system. They are mainly meant for digestive or lytic action. In case of endocrinal gland, the secretions are known as hormones, and these are having paracrine effect. They will also act locally and through blood, means as the secretion is poured into the blood, they are mainly regulated by pituitary gland, which in turn is controlled by hypothalamus, and the hormones regulate and coordinate the body function. There are many ways of classifying glands. First method of classification is based on the number of cells. So if a single cell is present, it is known as unicellular gland, and such cell may perform secretory function, for example goblet cell. Then multicellular glands have group of secretory cells. And second method of classifying glands is based on presence of ducts. So exocrinal glands have ducts to convey their secretions, and endocrinal glands are ductless and they pour their secretions directly into the blood. Based on the mode of secretion or manner of secretion, the glands can be classified as merocrine or epicrine gland, where the secretion is by exocytosis. Apocrine glands, here while secreting, the apical portion of the gland is shed off. Holocrine glands are those which when secreting will disintegrate the entire cell. Then based on development, glands can be classified as ectodermal, mesodermal and endodermal. The glands which are ectodermal in origin are of skin, mammary gland, lacrimal gland, salivary and pituitary gland. Mesodermal glands are suprarenal cortex, gonads, kidney and spleen. Endodermal glands are thyroid, parathyroid, thymus, liver, pancreas, prostate, urethral and bulbourethral gland. These are all endodermal in origin. We can classify glands as endocrinal glands which do not have duct in them. Exocrinal glands are those which have ducts in them. Exocrinal glands are divided into unicellular and multicellular. Unicellular are single cells performing function of secretion, and example is goblet cell. Multicellular cells are further divided by various methods into means there is further classification of multicellular glands on the basis of number of ducts. Now suppose a single duct drains the entire secretion of the gland, it is known as simple gland. And if there are more than one duct, so individual secretory unit has its own duct and they will ultimately drain into a single duct, that is compound variety of gland. So more than one duct will drain the gland. Then second way is on the basis of shape of the secretory unit. So if it is tubular, or it can be acinar or alveolar or flask shaped. So depending on the shape of secretory unit, it is classified as tubular, acinar and alveolar. Then on the basis of nature of secretory product. Now if the gland is secreting thin protein rich secretion, it is serous type of gland. If the secretion is very thick, viscid, mucus is secreted, then it is known as mucus variety of gland. And if there is mix means combination of both the secretion, it is known as mixed variety of gland. Then the fourth method of classification is on the basis of secretory mechanism, the manner or mode by which the secretion is poured out. So they can be classified as merocrine, apocrine and holocrine glands. We have already studied goblet cells in our previous lecture of epithelium. So the goblet cells are simple columnar epithelial cells which have glandular function and they mainly secrete mucus. So goblet means what? If the base is narrow and the apical portion is expanded, it is known as goblet shape. Then the mucus present in the apical part of the goblet cells in the membrane bound secretory vesicles is shed or it is washed off during routine H&E staining. So if we see in routine H&E stains, the upper part of the goblet cell which is expanded appears empty. Then the cytoplasm present in the goblet cell is pushed to the periphery by mucinogen droplets present in the secretory granules, and also the nucleus is shifted towards the base. Due to secretory function, the goblet cells have prominent Golgi apparatus and they have abundant amount of rough endoplasmic reticulum towards the base. The goblet cells are located in the epithelium of gastrointestinal tract and respiratory tract. Apart from H&E staining, the goblet cells can also be stained by periodic acid shift staining. It imparts magenta color to the mucin, and with the help of mucicarmine staining, deep red color is given to the mucin present in the goblet cells. Now it has got applied significance also. If the goblet cells, main function of the goblet cell is to protect the lining epithelium. So if the goblet cells are damaged, there will be defective mucosal barrier, and as they are lining the respiratory tract and the intestinal epithelium, so they will, if there is damage to the goblet cells, it will result in recurrent respiratory infection and intestinal inflammation. The first way of dividing multicellular exocrinal glands is based on the shape of secretory unit. So when the shape of the secretory unit is tubular, the tubule can be straight, coiled or branched, as we can see in the intestine, in the sweat glands or in the gastric glands. So in case of intestine it is simple tubular, and in the sweat glands we can see coiled, simple coiled tubular gland. Then the second variety is acinar gland. So here the secretory unit is spherical. Mostly we see acinar glands in the exocrinal part of pancreas and salivary gland. Then the third shape is alveolar. It is flask shaped secretory unit, and such alveolar glands are having distended secretory units. They are also known as saccular glands. Classifying multicellular exocrinal gland is based on the number of ducts that drain the gland. So if in case of simple gland, there are unbranched ducts. So the ducts do not undergo branching, and in compound glands there is branching of ducts. Now again, simple uh, glands which have unbranched ducts are further classified depending on the shape. So simple tubular, here a straight tubule is present. Commonly it's located in intestinal gland. Simple coiled tubular, here a single tubule is there and single duct is there draining the tubular part, and such coiled tube is seen in sweat gland. Then simple branched tubular means here the tubular secretory unit is branched, though a single duct is there. There are many secretory units, but all these secretory units will open in a single duct. So example is gastric gland. Then simple branched acinar. Again here there are branched acinar secretory. Many acinar secretory units are there, but ultimately all these acini will open into a single duct, and example is sebaceous gland. Now the second variety is compound glands. Here the ducts are branched. So again depending on the shape, there can be compound tubular. Here there are tubular secretory units, multiple secretory units are there, and all these units have their own ducts, and these ducts will ultimately open into a single duct. Example is Brunner's gland in duodenum. Then second variety is tubular acinar. So the secretory unit is acinar in shape. Again there are many such secretory units. They have their own branches, branched ducts, and these ducts will again open in a single duct. Example is exocrinal part of pancreas. Third variety is compound tubuloacinar. Here some secretory units are tubular in shape and some secretory units are acinar in shape. They have their own ducts, and again they will open in a single duct. Example is submandibular salivary gland. On the branching pattern of the ducts, exocrinal multicellular glands can be divided or classified into simple and compound. Simple glands are those which have a single duct which is unbranched. So there is no branching of the ducts. Example is crypts of Lieberkühn in the intestine, then the sweat glands, fundic glands of the stomach, mucous glands of urethra and meibomian glands. Compound glands are those which have branching of secretory ducts. Example is Brunner's gland, mammary gland and submandibular gland. We will see simple glands with unbranched ducts in detail. So the first variety of simple gland is simple tubular. Here the secretory unit is tubular in shape and it has a single duct. Example is glands of small intestine and uterus. Then the second example or variety is simple coiled tubular. Here the terminal part of the tube is coiled with a single duct. An example is sweat gland. So here we can see a single secretory unit and it is draining into a single duct. Then the third is simple branch tubular. Here the secretory part is divided into few branches, but all of them are opening into a single duct. So the secretory unit is tubular and it is branched, but a single duct is present. Example is gastric gland. Then simple branched alveolar, here the secretory part is divided into branches which are expanded and they form either alveoli, acini or saccule, and they open again into a single duct. Example is sebaceous gland. Compound glands are the glands which have branched ducts in them. So in the first figure we can see compound tubular gland. Here we can see branching and rebranching not only of the secretory part but also of the ducts. So each of the secretory unit has its own duct and such ducts will again open into a single duct. So example is Brunner's gland of duodenum. The first diagram shows compound tubular variety of gland which is seen in Brunner's gland in the submucosa of the duodenum. Then the second variety is seen in the second diagram. It is compound alveolar gland. Here the not only the secretory part is alveolar and it is branched, but such secretory part has again its own duct and the ducts are dividing into several orders. So example of compound alveolar gland is mammary gland. Then in the third figure we can see few secretory part is tubular while some of it is alveolar in shape, and again the ducts show branching in several orders. So example of compound tubulo-alveolar gland is salivary gland. Based on the nature of secretion, the salivary glands or the exocrinal glands are divided into serous, mucous and mixed variety of gland. So in this slide we can see serous glands. So the serous acini, the cells present in the serous acini are tall pyramidal. Nucleus is mostly round and in the center. If we see the staining of such cells, the basal part of the cytoplasm has more amount of ribosomal RNA. So it stains basophil with the hematoxylin. It appears more violet, and the apical part is eosinophilic because it has secretory zymogen granules and they stain with the eosin. And the secretions of the serous gland are mostly thin, protein rich, watery, and example of serous salivary gland is parotid gland. Depending on nature of secretion, the second variety of gland is mucous gland. In the mucous gland, acini are again tall columnar, nucleus is flat, it is pushed towards the base by the mucin or mucus present in the gland. Cytoplasm contains membrane bound droplets which contains mucinogen. So this mucinogen is washed off during routine HNA staining. So the mucus acini appear empty. So if we revise the appearance, the acini appear empty, then the cells are tall columnar, the nucleus is shifted towards the base by mucinogen, and such glands secrete thick viscid mucus. Example is sublingual gland. Mixed gland, some of the acini are having mucus secretion and some are having serous secretions. So some acini are entirely serous or mucus, or both the types of cells may be present in one acini. So the serous cells may present a crescent on the mucus acini, and example is submandibular gland. First diagram is of serous acini, second is of mucous acini, and third is of mixed acini. So if we see the serous acini, they are smaller in size compared to mucous acini. These are compound alveoli and the lumen of the serous acini is smaller compared to mucous acini. And the serous cells, these are pyramidal in shape, nucleus is round, it is towards the center. If we see the staining, in the basal part it is basophilic due to presence of ribosomes, and apical part stains with eosin because it has secretory granules, and the secretions are mostly thin, watery, enzyme rich. An example of serous acini is parotid gland. The second diagram is of mucous acini. So these acini are larger compared to serous acini. These are mostly compound tubular or tubulo-alveolar. Here we can see in the mucous acini, the lumen is larger and wider. The mucous cells are tall, columnar. The base is shifted towards the periphery. It is flat, elongated nucleus towards the base, and as the mucin or the mucus secreted by the gland is washed off during H&E staining, the acini appears empty. The secretions are thick, viscid, and example of mucous acini is sublingual gland. Third diagram is of mixed acini. So here we can see mucous acini and the crescent formed by serous acini. So this type of appearance is known as serous demilune. It is seen in mixed salivary gland, and example is submandibular gland. Serous cells are pyramidal in shape. In their basal part is basophilic because it has more amount of rough endoplasmic reticulum and ribosomes, whereas the apical part is eosinophilic because it has secretory granules which stain with eosin, and the nucleus is mostly round and present in the center, and the secretory granules have zymogen in them. And the second diagram is of mucous cells. So the mucous cells are tall, columnar, and their cytoplasm appears empty because during routine H&E staining the mucus is washed off, and the nucleus is flat, elongated. It is shifted towards the basement membrane by the mucinogen, and the granules, secretory granules, mainly have mucinogen in them. Depending on the nature of secretion, so far we have seen there are three types of acini: serous, mucous, and mixed acini. There are other varieties of acini also, like there is lipid or fat secreting acini. Here the alveolar cells in the lacrimal gland, mammary gland, or in the suprarenal gland, testis, ovary, they contain oil droplets and lipid droplets in their cytoplasm. So this fat gets dissolved in routine H&E staining, and if we want to visualize the fats, we can stain it with special stains like osmic acid where the fats appear black in color, and with Sudan 3 and 4 type the fat droplets appear red in color. So there are some acini which secrete lipid also. Then the other variety of acini which secretes steroids is characterized by presence of extensive smooth endoplasmic reticulum and prominent mitochondria. Now we know that either means those secretions which are rich in protein, in such acini there is more amount of rough endoplasmic reticulum, and in the glands or acini which secrete lipids or steroids, there the cells of such acini have more amount of smooth endoplasmic reticulum and they have prominent mitochondria. So apart from serous, mucous and mixed acini, we also have lipid secreting acini and steroid secreting acini. Steroid secreting acini are mainly seen in adrenal gland, and lipid secreting are seen in lacrimal gland, mammary gland, and the gonads like testes and ovaries. Based on the mode of secretion, glands are classified as merocrine gland, apocrine gland, and holocrine gland. So, in case of merocrine glands, the secretory products are discharged by the process of exocytosis. In such glands, the cells are not destructed and the secretion is poured by exocytosis. Example is pancreatic acini. Second variety is apocrinal gland. In apocrinal gland, when the secretory products are poured out, the apical portion of the cell will be shed off. Example is lactating mammary glands and apocrinal sweat glands and ceruminous glands. Then the third mode of secretion is holocrine glands. While discharging the secretion, the entire gland gets disintegrated by apoptosis, or it undergoes programmed cell death on maturation. And the example of holocrine gland is sebaceous gland and meibomian glands. When a gland or an acini or the cells in an acini secrete substances that affects the nearby cells, it is known as paracrine effect. So the secretions do not enter into the bloodstream, but through the paracrine effect, the secretions will enter into the extracellular matrix, and this will affect the target cells by diffusion process. And its example is vasodilators. So the vasodilators are released by endothelial cells of blood vessels, and once these are released, they cause relaxation of vascular walls. When the cells of an acini secrete a molecule that acts on the same cell, means the cell which is secreting, it will have the receptor for its own secretions, and these secretions will bind with the receptors present on the same cell. It is known as autocrine signaling. So it can activate positive or negative feedback. An example is activation of cells of immune system by interleukins. We have classified glands upon the nature, depending upon the nature of secretions, into serous, mucous and mixed. Also lipid and steroid secreting glands are there. All here we also have some glands which secrete cells as their secretion. So those glands which secrete cells as their secretion are known as cytocrine glands. Example is ovary, testis and bone marrow. So in the ovary, during the process of ovulation, the ova is shed off. In the testis also the sperms are released. So these are all examples of cytocrine glands. Malignancy of epithelial cell is known as carcinoma. Malignancy of connective tissue is known as sarcoma. Malignant growth in the gland is known as adenocarcinoma, whereas benign growth is known as adenoma.